In this episode of Let's Talk Toads, you'll learn how researchers use an experimentally adapted SCN collection.
Here we are at the Tode Farm at the University of Georgia. Behind me is the largest active collection of experimentally adapted soybean cyst nematode populations developed by nematologists dating back to the early 1990s. These include Drs. Victor Dropkin, Virgil Luders, Lawrence Young, Charlie Opperman, Prakash Areli, Terry Niblack, and myself. The collection represents the wide range of SCN HG types in growers' fields across the United States. These populations are used as workhorses for the soybean breeding program, as well as soybean researchers and nematologists throughout the United States. These populations have been adapted to reproduce on many of the current sources of resistance, including PI 88788, Peking, PI 90763, PI 437654, and glycine soja wild soybean. Some of these populations are adapted to reproduce on PI 88788. For example, this BD2 population here is highly adapted and can reproduce at greater than 50% female index on PI 88788. In contrast, the population here, MMBD3, is highly adapted to reproduce on the Peking type of resistance, greater than 50%. A major research breakthrough funded by the Soybean Checkoff was the sequencing of the soybean cyst nematode genome, which was released in 2019. With this genetic blueprint in hand, we now have the opportunity to exploit this rich collection of genetic resources to identify the genes SCN uses to reproduce on resistant varieties, that is, the SCN virulence genes. 30 years ago, we only knew that these genes were either dominant or recessive, that they could be inherited. 3 decades later, we finally have the genetic and molecular tools necessary to not only sequence the nematode genome, but sequence a wide variety of SCN populations that are shown here in this collection to compare their genomes to pinpoint the genes for virulence. The benefits are twofold. First, as we identify the virulence genes, we also identify key targets to potentially engineer novel forms of resistance to SCN. Virulence genes likely encode proteins found in soybean cyst nematode spit. By identifying the virulence genes, we can also develop a molecular test to diagnose a grower's field population and prescribe the most effective type of resistance to combat soybean cyst nematode. For prescriptive management to become a reality for growers, we need to integrate our knowledge of the genetics of plant resistance genes with the knowledge of virulence genes and soybean cyst nematode. Your soybean success starts here.